Taking on this assignment, I knew it would be both challenging and rewarding. Having grown up in rural environments, I spent a lot of time outdoors and in freshwater ponds. I chose to create an aquatic microcosm so I could learn more about aquatic microorganisms. I've spent multiple days working on this presentation so the viewer could easily follow along and want to see what happened. Enjoy. So my hypothesis, I had a null hypothesis, which was the aquatic microcosm will be able to sustain itself throughout the duration of the course. An alternate hypothesis, the aquatic microcosm will be able to sustain itself and organism productivity will increase. If done correctly, the microcosm will act as its own environment, meaning it will produce its own oxygen through photosynthesis, therefore promoting sustainability for all living organisms within. Then in 2007, I believe the microcosm will produce its own nitrogen cycle, just like planet Earth, Galileo 2004. So I wanted to do an aquatic microcosm because I would have a better chance at seeing organisms, and that's what Professor Whitworth suggested. I located a small retention pond just outside of where I work. It appeared to be a failed drainage system, but lucky for me, it held water. The water was extremely clear and even had a decent number of green algae. I knew this was my area to gather. The steep rocky sides proved challenging to navigate, but with a little luck and determination, I gathered nearly 1 gallon of water, a handful of green algae, and 2 cups of sediment. Upon gathering materials for the microcosm, the water was very murky. No light could pass through the water, no heterotrophs could be seen, and the algae introduced was not visible. I knew I had to be patient and wait for the water to clear. Once I got home, I placed it in a bay window on a black chair near a heater vent. I wanted to receive maximum sunlight and proper heat from the black chair and vent. At this time, or as time went, the water cleared up, the algae expanded, and seemed to spread through the container. And I began to see heterotrophs swimming around. I noticed what happened, what appeared to be nematodes, flagellates, copepods, roundworms, and what appeared to be water mites, but they're too small to see their features. Illustrations retrieved from msnucleus.org. Some of these organisms are actually parasites, and one of the reasons drinking stagnant water will make you sick. Zimmerman 2020. The chart follows a relationship between the clarity of the water, heterotroph count, and autotroph count. I found it interesting that the clearer the water became, the more life was present. Not only visible, but more organisms were observed swimming around as if the oxygen levels in the water were rising or food was becoming more abundant. Another explanation could be that animal productivity was also on the rise. Although the algae started to sink towards the bottom, it continued to produce oxygen as seen in this video. Condensation continued to build within the container, and air bubbles would still come from the sediment and rise to the surface. I believe my hypothesis was correct, as life did appear to be sustainable. The ecosystem was producing its own oxygen, therefore feeding the heterotrophs within. I did not witness any omnivore or carnivore activity, but that's not saying it did not happen. I'm still not sure why the algae started to settle towards the bottom. When I pulled it from the water hole, in the beginning, the algae was in long strands and appeared to be connected to the sediment below. The algae mimicked this in my microcosm for the first 4 weeks, then started to sink. The skies were cloudy for a few weeks, and it did get colder outside, so maybe that's why. For further research, I'd like to be able to test the temperature of the water and record it daily. Water temperature is likely a variable that I did not capture. Next time I'll leave more air on the top of the container. This will replicate the atmosphere more closely. If I had the equipment, I would also add a heating source, more stable and constant than the air vent I used. Microcosms are primarily used to study the interactions between 2 species. Ives, 1996. While our projects allowed this, there's more that can be observed. Although my microcosm was aquatic, it held similar characteristics to that of the boreal forest. The algae that floated at the surface acted much like a forest canopy. It provided oxygen through photosynthesis and a food source for herbivore creatures. Chapman Bowl in 2015. Biodiversity of organisms could be an issue in microcosms. Without a microscope, it's difficult to tell if there are missing animals that would have been variable from where I collected the sample, meaning I did not collect the flying insects that lay their eggs in the water, or a mammal that drinks the water and introduces more variables. Independent and dependent variables have to be taken into consideration when collecting materials for a microcosm. The researcher needs to be sure to collect the same materials that are present in nature so the results can be as close to true as possible. Adaptation is a desirable observance in nature. Adaptation within the microcosm is unlikely, as adaptation usually takes multiple generations to accomplish. Bach, 1965. Although microcosms could last decades, it's not a guarantee. References cited, and thank you for viewing my project.